Welcome to this explainer. Let's just dive right in, shall we? Today we're looking at nucleic acids, but we're going to do something a bit different. We're taking a bottom-up architectural approach. So, instead of just throwing the famous double helix at you right from the start, we're going to build your understanding step by step. We'll start from the absolute smallest molecular pieces and assemble them all the way up to the complete molecule.
So here is the game plan for how we'll construct this knowledge together. 1. The blueprint of life. 2. Meet the nucleotide. 3. The genetic letters. 4. Building the double helix. And 5. From structure to function.
All right, Section 1, the blueprint of life, understanding the archive. Because before we start building anything, we got to know what we're actually making.
So, what makes you, you? It's a pretty heavy, profound question, right? But down at the molecular level, the answer is actually right in front of us. It's nucleic acids. These are massive biological polymers, and they're basically the ultimate information managers inside your cells. They store and transmit your genetic code. And there are two main players we care about here: DNA, which is your permanent archive, and RNA, which is the working copy that actually executes those instructions.
I actually love this analogy for keeping them straight. Think of DNA like a master original cookbook. It's kept under strict lock and key in this grand library, which in your cell is the nucleus. You would never, ever risk ruining the original book, right? So when you actually want to make a meal, or in the cell's case, build a protein, you just make a photocopy of one single recipe. That short-lived working photocopy, that's RNA. You carry that out into the messy kitchen to do the actual cooking.
Let's move right into Section 2, Meet the Nucleotide, the building block. We're zooming all the way in to see how these massive archival molecules are built from scratch.
Every complex structure has to start with a simple brick. And in nucleic acids, that brick is the nucleotide. This is the single repeating monomer unit. You link them together over and over and over, and you get a nucleic acid polymer. And fun little side note here, even when they aren't chained together, individual nucleotides can actually act entirely on their own as vital cellular energy carriers. ATP is a perfect example of that.
Now, a nucleotide isn't just one solid blob. It's actually made of exactly 3 components. If we look at it from left to right, first you have a 5-carbon sugar, second a phosphate group, and third a nitrogenous base. The really crucial takeaway here is that you absolutely need all 3 of these to have a nucleotide. If you snap off that phosphate group, it completely changes its chemical identity. Chemists actually call that a nucleoside instead, so the phosphate matters.
Remember earlier how we said RNA is the working copy and DNA is the master archive? Well, that functional difference starts right here, at the monomer level. The specific identity of that 5-carbon sugar is what fundamentally separates these two cousins. DNA uses deoxyribose. Deoxy meaning it's literally missing an oxygen atom at the 2' carbon. That missing oxygen makes it highly stable, which is perfect for long-term, double-stranded storage. RNA, on the other hand, uses ribose. That extra oxygen makes it a bit less stable, meaning it's usually single-stranded, perfectly suited for being a short-lived messenger.
Okay, Section 3, the genetic letters, the variable bases. So the sugar and the phosphate, they're always the same, but that base, that can change. And that variability is literally where the magic happens. Because it's the only variable part of the nucleotide, the nitrogenous base is the actual letter that encodes your genetic information. Chemically speaking, these bases fall into 2 structural families. First, you've got the purines. These are larger double-ring structures, specifically adenine and guanine. Then you have the pyrimidines. These are your smaller single-ring structures: cytosine, thymine, and if we're talking about RNA, uracil.
Now, it can be totally annoying to remember which base belongs to which family, but here is a brilliant little memory trick for you. Just remember the phrase pure as gold. The purines are adenine, the A, and guanine, the G. Pure as gold. If you remember that, then you automatically know everything else. Cytosine, thymine, and uracil must be the single-ringed pyrimidines.
So these letters don't just float around randomly. They pair up to hold the DNA strands together. Adenine always pairs with thymine, using 2 hydrogen bonds, and guanine always pairs with cytosine, using 3. But why? This is honestly one of the most elegant things about molecular chemistry. It is purely geometric. A large double-ring purine absolutely must pair with a small single-ring pyrimidine. Why? To keep every single rung of the DNA ladder exactly the same width. It is just perfect chemical engineering.
All right, we have our pieces, we have our rules. Let's get to Section 4, building the double helix, snapping the pieces together.
To actually build the chain, the phosphate of one nucleotide chemically links to the sugar of the next one down the line. This creates a really strong, repeating sugar-phosphate backbone. Now, because of exactly how the carbons are numbered on that sugar ring, the chain ends up having a very specific direction. Chemists read it from what's called the 5' end down to the 3' end. You can think of these backbones as the solid outer rails of a ladder, while all those nitrogenous bases point inward to meet their partners.
And here is the grand reveal, the architectural wonder itself. When two of these long strands finally meet, they form the iconic double helix. But pay close attention to how they align. The two strands are antiparallel. That means they run in opposite 5' to 3' directions, kind of like traffic flowing on a two-way street. Bound together by those perfect AT and GC pairs on the inside, the entire molecule winds clockwise into a right-handed twist. It's a stunning structure, famously pieced together by Watson, Crick, and Franklin.
So, we built it, but what does it actually do? Section 5, from structure to function, the molecule in action.
Because of that perfectly geometric matching system we just talked about, DNA can seamlessly and rapidly copy itself in a process called replication. The two strands literally just unzip down the middle, and brand new complementary nucleotides float in and match up to each side. This guarantees that right before a cell divides, every new cell gets a flawless, complete copy of your entire genome.
Now, when it's time to actually use those instructions to build something, we enter the first step of gene expression, which is transcription. The DNA safely opens up just a little bit inside the nucleus, and its code is carefully transcribed into that short-lived working photocopy we talked about way at the beginning, messenger RNA, or mRNA. Once that's done, the messenger RNA leaves the library and heads over to the ribosome. This triggers translation. The ribosome, which, fun fact, is actually made of ribosomal RNA itself, reads that mRNA code. Then these tiny little delivery molecules called transfer RNA bring in the exact matching amino acids to assemble a complete functioning protein. So the structure of the molecule directly enables its expression.
But nature isn't always perfect, is it? Sometimes during that replication process, a chemical copying error happens. An A might accidentally slip in across from a C. This is a mutation. Now, normally we hear that word and think it's a bad thing, but these occasional molecular typos introduce genetic variation, and that is the absolute vital ingredient necessary for populations to adapt and evolve over time.
Okay, let's pause for a quick knowledge check. Let's see how you do. If a DNA strand reads 5' A, T, G, C, 3', what is the complementary sequence? Think about those base pairing rules, and, critically, think about the direction the new strand has to flow.
Did you get it? The answer is 3' T, A, C, G, 5'. Adenine pairs with thymine, guanine pairs with cytosine. But the real trick there was remembering to flip the numbers. The complementary strand must run antiparallel, so it has to start with 3' and end with 5'. If you caught that, huge congratulations. You have completely mastered the geometry of the double helix.
Let's do a super quick recap of our bottom-up build today. We started with the basic nucleotide, a sugar, a phosphate, and a base. We split those bases into purines and pyrimidines. We saw exactly how a large and small base perfectly pair up geometrically. We linked them into antiparallel strands to form the double helix. And finally, we saw how that specific elegant shape makes replication, transcription, and translation possible. To wrap up this explainer, I want to leave you with one final thought to chew on. We just spent this entire time marveling at the perfect rules of DNA, how A must pair with T, how the latter is a constant width, how the transcription is so precise. But if copying errors, mutations, technically break these perfect, beautiful rules, how is it that those molecular mistakes are actually the vital raw material that drives the magnificent diversity and evolution of all life on Earth? Thanks so much for joining me on this explainer, and keep questioning the chemistry around you.